reality that we're living in right now is that we do live in this COVID world. And we are certainly proud of all the efforts that are being done from all of you that are on for this annual member meeting that has had some bumps along the way. But can you hear me okay?
I would just wait a second for people to join.
Oh, okay. So now I'm being told I should wait a little bit longer for people to join. I'm going to keep going. Anyway, again, very proud of everything everyone's doing from on the PPE front to the testing kits to therapeutics, as well as with patient care. So a key driver certainly in controlling this crisis from happening in the future is having vaccines. We have two companies talking today about their efforts to advance their vaccines through clinical trials in record speed so that we have safe and effective vaccines for COVID-19. So first we'll be hearing from Dr. Nettles of Johnson & Johnson, followed by our Wisconsin company, FluGen, which will be Dr. Eiden. I will not be reading any bios, which are on our main page. And a reminder, if you have any questions, please email Mike Harrison, mharrison@bioforward.org. With that, Dr. Nettles, let's have you lead off and tell us about what Johnson & Johnson's efforts.
All right, thank you so much for inviting us today. We're the same way at J&J. We're fully virtual, so I feel your pain when we're trying to get these virtual calls up and running. It's almost an hourly occurrence, but thank you for inviting us to talk about our R&D effort in response to the COVID-19 outbreak. If you can go to the next slide. Most of what I'm going to talk about today has to do with our research and development response, but I do want to take a second to just acknowledge and thank the frontline healthcare workers, the essential workers, and acknowledge the tremendous loss and recognize those who have lost members of their family due to this outbreak. In addition to the research and development effort, J&J has tried to step up and be of support in a number of other ways. Since the beginning of the outbreak, we have taken measures to make sure that we have a secure drug supply and tried to eliminate the occurrence of shortage of any medications that are not related to COVID-19. We've taken steps in some instances to alter our production paths. So for instance, in Nettles, Pennsylvania, we altered a Listerine production site so that we could make hand sanitizer in order to supply that hand sanitizer to those who were doing the essential work working on our vaccine at J&J. Another example, J&J allowed healthcare providers like myself to take leave from their job at J&J to go back and care for individuals who are impacted by the COVID-19. So our chief medical officer of our consumer business has spent time in the emergency room in New York City caring for patients. So those are just some of the examples of ways that J&J has been trying to step up in this instance. But if you go to the next slide, with regard to the specific research and development aspect, J&J has taken a two-pronged approach. One is collaborating in an attempt to develop a safe and effective vaccine. And the second is screening our late stage and marketed compounds to try to identify compounds that may have antiviral activity or treatment effect in those who have been infected with coronavirus. So let's go to the next slide. So now we're talking specifically about a preventative COVID-19 vaccine. And here it gives you a timeline of what has happened so far this year and our aspiration that lead us to the beginning of next year. So as you know, with the start of the outbreak in Wuhan, China in late 2019, an important event was in early January, there was publication of the virus sequence, which allowed us to understand the genetic code of the coronavirus. At Janssen, we began working that same month on vaccine development, recognizing that this had the potential as a global pandemic. We announced that we had selected our lead candidate for development of our vaccine in late March. We anticipate that we'll be in first-in-human clinical trial at the latest by September of this year. We would then have interim phase 1 study results late in 2019. And our aspiration is to be able to supply our vaccine as an emergency use authorization in the US, much like remdesivir is being used right now, early in 2021. So let's go to the next slide, and I'll talk a little bit about our vaccine approach. So many of you would recognize the developmental pathway for medications and for vaccines, and that's shown on this slide. So typically this process would take approximately 10 years, but there are examples of important vaccines which have taken more than 20 years in development. So you can appreciate the extraordinary efforts that are being taken to try to develop a vaccine really in this 1-year time frame. The usual developmental pathway has us selecting a lead compound, moving through phase 1, 2, and 3 clinical trials, where we move from small number of patients in the phase 1 trial, then moving to an intermediate number of patients and a large number of patients in phase 3. And after that, we really focus on the scale up of the production of the vaccine. After you know that you have an effective vaccine, what you're seeing in this situation is we're doing a lot of this rather than in sequence, we're doing it in parallel. So before we've started our phase 1 clinical trial, we've also begun to try to find ways to ramp up our manufacturing of this vaccine so that it will be available as soon as we have a read on the efficacy of the vaccine and the safety. Let's go to the next slide. This is a little bit about Johnson & Johnson's vaccine approach. What you're going to see is that there are over 100 vaccines that are in development against the coronavirus right now. And that's really fantastic news, because what the truth is right now is we don't know which vaccine approach is going to give us the best effect. And so we need to be moving forward with multiple vaccines from multiple companies at the same time. And we need a couple of those vaccines to make it to the finish line. What I'm showing you here on this slide is Janssen's approach. We are taking an approach with a vaccine platform that we have been using to develop vaccines against other very serious infections, including HIV, RSV, which is a respiratory infection, Ebola infection, and Zika. So we've used this platform against those infections. We've had now over 60,000 individuals vaccinated with this platform that we're using, and it's generally safe and well tolerated. So that gives us some confidence that we can move forward with this platform in a vaccine development against the COVID-19. What the vaccine is that it's actually an adenovirus. That an adenovirus causes the common cold. We've taken that adenovirus and taken out a portion of the gene of the adenovirus that makes the adenovirus unable to copy itself or replicate or make you ill when it's injected into you. And we've put into that adenovirus a piece of the coronavirus that is aimed at the spike protein that you've probably heard about on the news. And so then you actually use the adenovirus to inject a piece of the coronavirus into the individual. And the hope, as is with all vaccines, is that this provides exposure to a piece of the coronavirus in your body, and your body mounts an immune response against the coronavirus. The other advantage that the J&J platform has right now is that this vaccine is being produced in a Percy-6 cell line, which is a cell line that allows us to produce large volume of the vaccine, so many, many doses of the vaccine in a relatively small footprint. So taken together, this adenovirus and Percy-6 technology, we feel is an important contribution to developing a coronavirus vaccine as fast as possible. So let's go to the next slide. So based on those capabilities that I've just described, the goal for Johnson & Johnson is to produce up to 1 billion doses of a safe and effective vaccine, and we will keep producing them if more are needed after that. So next slide. In addition to the production of a vaccine, we've been working on trying to find treatment solutions for coronavirus. So a vaccine is for somebody who's uninfected, and you use the vaccine to try to prevent you from becoming infected. But what we also need is treatment solutions for individuals who have already been infected. And so what we have developed is a cell line, and we're screening our compounds to see if they have antiviral activity and could be used as treatment. This effort has kicked off in a similar timeline as shown on the prior slide. And we're moving forward by screening our compounds that are currently marketed right now. The advantage of that is we understand the safety profile of those drugs, and we have a large drug supply already available so that they could be used. After the screening of the currently marketed products, we're going to screen the products that are in phase 3 and phase 2 development, and then we'll move through our antiviral library that are in earlier development after that. So let's go to the next slide. It's important really to say that none of this work that I've just been speaking about could be done without collaboration. And Janssen is really looking to be a collaborator in this situation. So much of the funding of the work on the vaccine and the drug screen has been done in collaboration with BARDA, which is an arm of the federal government that is built to deal with pandemics or disasters similar to this. The vaccine has been done in collaboration with Beth Israel Deaconess Hospital, and the drug screen with the Rega Institute in Europe. It's important to point out that scaling up our production of the vaccine is just as critical as finding an effective vaccine. And J&J is looking to find collaborations around the world. Our initial production site will be in the Netherlands, but we have two collaborations in the US with Emergent Biosolutions and Catalent Biologics to be able to produce the necessary drug supply. And we're looking for other collaborations in Asia as well. Next slide. So, sometimes it's hard to be a big company, and to be a biotech, to be able to really move nimble is desirable. But in other instances, such as a pandemic like this, there really are advantages to being a large company like J&J, to have that global reach, to have that over 130-year advantage. We've been here through prior pandemics. We feel like it's a commitment that we find ways to contribute to ending this pandemic, and we intend to be here for future pandemics as well. So I'll stop there, and we can take any questions now or later in the conversation.
That'd be great. Yeah, I was thinking after Dr. Eiden presents, then let's have some questions. So next up, our local company, FluGen. Dr. Eiden, we'll hand it over to you for you to present on FluGen's efforts.
Thank you very much, Lisa, and Rick, thank you for that excellent talk and presentation, and I very much appreciate the discussions. I think we're working backwards. This is my ending slide. There you go. Okay, thank you. I'd like to thank BioForward and the group for the opportunity to present on behalf of FluGen and collaborators. And what will be described in the next several slides is development of an influenza vector COVID-19 vaccine. Next slide, please. As Dr. Nettles has noted, there are numerous vaccine efforts underway now, which I think we all very much appreciate that so many researchers have participated in this activity. It's worth noting that not only are there well over 120 individual vaccine efforts, but these fall into multiple categories for types of vaccines. So it's not just multiple shots on goals, it's multiple ways of getting there. Here in this slide, I think you'll recognize a number of programs that have been in the news lately, including J&J's adenovirus vectored vaccine, as well as the Moderna mRNA vaccine. Pfizer also has a program in that category. And Novavax has received a lot of prominent notice over the last several months with their inactivated vaccine approach. I'll be discussing today the vaccine at FluGen, which is listed at the bottom here, and is a live intranasal vectored vaccine. And I'll explain all that in the coming slides. First slide, next slide, please. The technology for this approach with influenza vaccine is based upon inventions and innovations from Dr. Yoshi Karaoka's lab at University of Wisconsin-Madison, along with his teammates and collaborators, and focuses on the M2 transmembrane protein of influenza, which is an essential vaccine component necessary for replication. The vaccine is produced by preventing M2 expression and therefore preventing influenza replication, in this case by deletion of a segment of the transmembrane portion of the protein and 2 stop codons. Next slide, please. The vaccine is produced in a virocell line that constitutively expresses M2. So even though the influenza vaccine itself lacks M2 and cannot replicate, the vaccine expresses M2 from the virocell line and is able to undergo a single round of infection in humans and is administered, as shown in the bottom left of this slide, by intranasal inoculation. Now, as many of you know, influenza, shown in the top half of the diagram, undergoes multiple cycles of virus replication and causes disease, which may be mild to very severe or resulting even in death. But for most folks, this results in protection, imperfect, but good protection against influenza subsequently over many seasons, and it's believed for at least a decade. And we have data that it can actually last much longer than that in some circumstances. So an approach that uses an M2 deletion with single round replication avoids the illness that is characteristic of influenza, but is also able to induce a robust immune response very much parallel to natural infection. And as you'll see in one of the slides that comes a bit later, it's able to protect against influenza infection in a human challenge model. Next slide, please. Importantly, with respect to an intranasal inoculation with a live virus, vaccine in this case, one sees induction of all major immune effector systems, including innate immunity, mucosal immunity, cellular immunity, as well as humoral immunity. For influenza, humoral immunity is probably the most recognized. All the others come into play. Preclinical studies have shown with the M2SR vaccine from FluGen that unlike licensed vaccines that are given intramuscularly against influenza, the FluGen vaccine is able to induce each of these components of the immune system, not just the humoral immunity, which is characteristic of inactivated intramuscular vaccines. Next slide, please. In a human challenge model, a bit over 100 volunteers at the SGS site in Belgium were randomized to receive either placebo or a monovalent H3N2 M2SR vaccine for influenza. And after a single inoculation intranasally, these adults were then challenged with live influenza and followed for both infection and infection with illness. And the results of this challenge study for the placebo group who did not receive vaccine but did receive a challenge is shown in red, with almost 75% of those individuals exhibiting infection following challenge and approximately 50% exhibiting infection and illness. The volunteers who received the M2SR H3N2 vaccine had decreased infection as well as infection and illness. And most prominently, as you can see in the dark blue bar on the right, individuals who responded to the vaccination with a serum microneutralization titer against the challenge strain had an even more dramatic benefit for this vaccination. It's of note that this particular challenge study differed from any other that we're aware that's ever been performed in humans in that the challenge strain was highly drifted compared to the vaccine strain. So there's almost a 9-year difference here. That 9 years required 4 different vaccine changes during that intervening period to maintain vaccine efficacy with licensed vaccines. So here we're looking at a result where the vaccine itself is greatly distant from the challenge. And normally one would expect with licensed vaccine very little, if any, benefit from this. And here we're seeing a very positive result in prevention of infection and infection with illness. Next slide, please. So one of the things we wanted to build on at FluGen after seeing this results was to ask, well, we gave a dose of 10 to the 8th TCID 50 for that challenge study, and we got benefit for some of the people who responded, but we didn't get it in everybody. So how could we improve on this? The next step, therefore, was to go back and look at increased dose levels of M2SR vaccine and see if we can improve upon the immune response that was induced by the dose that was used in the challenge study. And in this case, we're showing results from a phase 1 study that was completed earlier this year that included a dose level of 10 to the 9th TCID 50. And the results, again, are rather dramatic, looking at serum hemagglutination inhibition responses, which is the correlative protection often used for influenza vaccines. The results in green are the results in green are the 10 to the 8th dose that was used in the challenge study, and the results in blue are from the 10 to the 9th dose in this phase 1b study. And as you can see, this increased dose results in a dramatic increase in the very serum assay that is the recognized correlative protection for an activated vaccine, something that's not normally seen with live intranasal influenza vaccine. Next slide, please. Safety is also an important component, as well as reactogenicity. One of the reasonable things that we should always ask with increased doses is, do we have a safety profile that will allow us to go forward? In this case, the answer is yes. Shown here are signs and symptoms in volunteers during the 7 days after vaccination with placebo, 10 to the 8th or 10 to the 9th doses of the M2SR influenza vaccine. And as you can see, while there is an increase relative to placebo, it's not a great increase. And there's really not much difference between 10 to the 8th and 10 to the 9th doses. Next slide, please. Looking further at comparing these immune results with this vaccine against live attenuated influenza vaccine, one can see here with the M2SR vaccine in the right-hand column and the licensed FluMist vaccine shown in the middle column, that both in measurements of GMT and gene fold rises in titers against the HAI correlate in adults, especially those who are seronegative at baseline. There's a rather dramatic difference between the single round M2SR vaccine that was used in the study, 71% seroconversion rate, which is in the same ballpark as one would see in similar patients or populations with live intramuscular vaccine, much greater than that seen with FluMist in adults. So this gave us very great hopes of moving forward, and we think these are very positive and encouraging results on the influenza vaccine that is being developed at FluGen. Next slide, please. So in summary, we've shown in preclinical models and with data that I have not presented here with human studies that all major forms of immune effector systems in humans are engaged in immune responses are seen with M2SR. But there's also a very positive result and very encouraging humoral immunity in the same level approximately that one sees with licensed influenza vaccines. Next slide, please. This then gives great encouragement for using this influenza live intranasal vaccine as a platform for a coronavirus vaccine for prevention of infection with COVID-19. Next slide, please. And the way this is approached is to insert into the live intranasal vaccine sequences from the spike antigens of COVID-19 and have those expressed on M2SR. One now then has a vaccine shown on the right that is expressing not only key components of influenza, in this case, hemagglutination and neuraminidase antigens, but also COVID-19 antigens, but is limited to a single round of replication and therefore not able to cause disease. Next slide, please. So going forward, the technology for this was developed at University of Wisconsin, again, in Dr. Yoshi Kawaoka's lab with Dr. Gabby Newman and their team. Production of vaccine and the components necessary to move forward with a vaccination program was at FluGen. And FluGen has now developed a collaboration with Bharat Biotech, a very large vaccine producer in India. They have developed over 15 different vaccines for market, including an H1N1 inactivated vaccine, as well as a number of other prominent vaccines, including live rotavirus vaccine. Tech transfer is underway from FluGen to Bharat in India, and Bharat will be scaling up and manufacturing vaccine and taking the lead on the first clinical trials with this live intranasal vaccine based on an influenza M2SR approach. And the first clinical trials are expected to occur in the third quarter of this year. Next slide, please. Next slide, please. I'd like to be sure to recognize the collaborative work with development out of the labs at the University of Wisconsin, as well as the work at FluGen under direction of Dr. Pamuk Bilsel, the Chief Scientific Officer, and Renee Huber in particular, who's been responsible for the clinical trials, the data of which you've seen for influenza, and for directing vaccine production, and recognizing the partnership with Bharat Biotech, with founder Krishna Ella and Dr. Rosh's Ella, who is head of development there. Thank you very much, and we'll be pleased to answer any questions you might have.
Great. Thank you so much, Dr. Eiden. So, question I have is, how will vaccines change the course of this disease? And what will happen if people don't get vaccines? Because that also has been the news. Government can't tell me what to do, but we also know just from history how important vaccines have been, obviously, with measles, smallpox, your polio. So could you just describe what the impact will be when we do have vaccines available for COVID, what this will do to get us out of this crisis and to stop this pandemic? Dr. Nettles, do you want to start? Okay, either one.
Go ahead, Rick.
Well, you know, in the event that we have a safe and effective vaccine, I think it's going to have a major impact on the pandemic. You know, your question hits a little bit at vaccine hesitancy, which is certainly a big issue in the U.S. And, you know, the balance between personal liberty and the safety and well-being of the population is certainly on full display on every night of the news here in Pennsylvania, just like it is there, I'm sure. And so it is a really complex issue. But you know, having that safe and effective vaccine would really make a major impact in reducing the burden of the diseases. And you know, we live in very interesting and uncertain times right now, where you could be in situations where institutions may ask for you to show them proof of having been vaccinated before you would be allowed to enter. I don't think any of those things are outside the realm of possibility over the next year. None of us can really say, but I think, you know, the vaccine really is our biggest hope to really getting out of this crisis that we're in right now. That's my perspective.
Dr. Eiden?
I totally agree with Dr. Nettles. I can add a few extra notes. I think some of the issues will be also related to the level of efficacy and the type of efficacy that is achieved. And we can see the distinctions with vaccines that are currently widely used and with various results. On the one hand, I can remember when Haemophilus influenza B conjugate vaccines were first introduced and widely used. As an attending physician, I don't believe I ever had a week on my service where I did not have a baby with Haemophilus influenza B disease. 2 years after that vaccine was out and widely used, we never saw it again. I remember the last patient I saw with Haemophilus influenza B vaccine, and when I took my students and residents around, I said, I really want you to know this disease and this patient who happened to be in the ICU, because I don't think you're going to see it again. And that's with a vaccine that is recommended and highly used. On the other hand, we have influenza vaccines which do provide efficacy and are safe, but which are imperfect. And in that case, at best, with H3N2 with licensed vaccines that we have today, you achieve 50%, maybe 60% efficacy. It's one of the reasons FluGen is working on an influenza vaccine as well as developing a COVID-19 vaccine. Nonetheless, we want everybody to get that influenza vaccine that's out there, because it provides benefit, and the biggest benefit is seen in severe disease. And as Dr. Nettles was saying, I think even today, we like to be sure that our healthcare workers are vaccinated. It's not always easy to do that. We often feel fortunate to get 70% compliance on the healthcare workers, but we know it provides vaccine positive effects not only for the healthcare workers themselves, but also their patients. So I think as we begin to see safe and effective COVID-19 vaccines coming forward, you'll be looking for protection against high-risk groups, the elderly, those with complicated medical conditions, but also there'll be great benefit to healthcare workers, and that will not only go for themselves, but for their patients. And as Dr. Nettles and I were exchanging a bit before this began, we were saying, what would it take for all of us to be back on an airplane? I think a safe and effective vaccine would allow us to begin thinking about public transportation, restaurants, all these things which have great impact on society. So it'll change a lot. It'll change a lot.
So Dr. Nettles, we have a question from the audience. And again, you showed that timeline where the normal vaccine process, I mean, it can go up, I think you even had it up to 22 years or something. It's a long time to really test safety and efficacy, right? So can we really test a vaccine this quickly to then get out to millions, billions of people? Is this doable considering what we've always done in the past just to have that safety and efficacy? Because we've just squished those timelines. Is it realistic that we can?
Yeah, it's the critical question. And so I know at J&J and the other manufacturers, I'm sure just the same way, safety has to be the top priority here. And so we really have to be sure that the vaccine is not going to induce some situation where we make the disease worse in either a small number of individuals or in certain types of individuals. The most important things that we're doing at J&J is we're using a platform, and it sounds like the platform that was just described using the influenza vaccine, using platforms that have shown themselves to be safe in other situations and making small modifications to those platforms. And the other way that you can try to accelerate things are based on those collaborations that I was describing. There's never been a situation that I've seen in my career where people are willing to review a protocol and provide you feedback from the FDA within a day or two. So it's trying to cut out those times that aren't really necessary having to do with the clinical trial, but more have to do with what it used to take to get review of a protocol and get a protocol up and running. Now protocols are getting up and running in a number of days because of the urgency of the crisis here. So we need to cut the timelines there, cut the timelines like I described on producing the vaccine in parallel with running your clinical trial rather than doing it as a serial situation. Those things I think we can cut in a safe way. And you'll see the phase 3 trials that come out from the manufacturers later this year are going to be very, very large. So you won't see us cutting the number of participants in order to move quickly.
So another question from the audience was, what is the expected timeline following demonstration of an effective vaccine in adults to have this offered to children or pediatric populations? Dr. Eiden and Dr.
You want
Nettles?
to take a first shot at that one?
How much longer, is that time span usually that you're starting to try and put this into pediatric or young children?
I think once you've got safety in adults and the vaccine is showing promise, it will very quickly go into children as well as the older age group. And in this particular case, that older age group is of great focus because that's where much of the mortality is coming. And I think that having that safety database, a large safety database quickly in younger adults will be key to both of those populations. And my personal expectation would be it will very, very quickly move down. It'll have to be done in small numbers initially. You're working with very vulnerable populations, so you'll want to take great care with that. But I think that can also move fairly rapidly in this case.
Yep, I would agree.
So Dr. Nettles, I think you addressed a little bit of this, and I think what I'm seeing, we're FluGen has now partnered with biotech to manufacture. One of the questions we had is, once a vaccine is developed and approved, what challenges do you face to scale up and provide a vaccine to a global population in a timely manner? Part of this is you already have scheduled your manufacturing, right? I mean, you're jumping ahead before you even get those clinical trial results back, right? Is that, or is there more to this whole scale up? Maybe that I'm missing, but that was really interesting, where manufacturing is already starting, or that's how you're partnering, which is very different from past how you normally do vaccines.
Yeah, that's right. And we talked a little bit about the different types of vaccines. Some are more difficult than others to scale up. So it really depends upon which type that we find is most effective and safe here on how hard they are to scale up. Some are easier than others to scale up. But yeah, just making sure that you have that space so that they can begin to work with you in a timely manner. And then the last piece is going to be, once we have the volume of vaccine, is how we distribute it. And there's a lot of complexity there, but partnering with the federal government, state and local health authorities to make sure that we can get the vaccine once we have it to enough people in the population is going to also be a challenge that needs to be worked through over the next 6 months.
Yeah.
So, so part of this testing then also is how many doses, right? So as you do this testing, you're determining, is this a single dose, whether it's nasal or into the muscle, or, I mean, how soon do you understand that? And again, I've never done this kind of testing. How soon do you know that it's going to either take just one shot or one nasal spray versus multiple? Does that come out pretty quickly in phase 1, or is that a phase 2, phase 3?
That's usually just identified fairly quickly, because you're going to, as I think you've seen from the group in Oxford that are also using a vectored vaccine, and they've announced both 1 and 2 dose results in their earliest study. So very early on in clinical development, phase 1, phase 2A, you can look at multiple doses. The other question that comes up is, how long does the protection last? Mm-hmm. And that also becomes extremely important. You build that in early during the program. Usually there's enough time here, it's going to go much quicker, but you'll want to know, okay, is this a vaccine that's going to provide 12 months of efficacy? Is it something that's going to last for several years? When do you need to give a booster dose? So following those first cohorts of volunteers who are in studies for longer periods of time, following their immune response, how it decays, and then building in how to look for booster dosing. We know, for example, with influenza, that the data are suggesting that by 6 months, many, if not most people who receive influenza vaccine have markedly diminished protection. And that's one of the reasons why we give yearly vaccinations. The other reason is the antigens change. So you can have strain changes or even shifts with more dramatic changes with influenza. But the duration is another very important component that relates to whether you are going to need a booster, and that therefore gets back to production. So that you're adding on, especially if you're looking at potentially vaccinating the entire world's population, but say the vaccine has to be a booster dose within a year. Now you've got this issue about getting first doses out to a population versus using your production to give booster doses. So there's a lot of complexities that are being anticipated. I think you've got some of the very best public health groups in the world focusing on these types of issues and working with manufacturers to address these.
Great. So I know we're getting to the end, but it was something that Michael Chambers of Aldevron said. So I just want to follow up on his comment. I think he's saying, this has been terrible on all of us, but also a lot of good can come out of something like this, right? We behave differently, and how even the biopharma industry has reacted to this. Is that, can this even be used for other disease states to move more quickly? Are there ways that we can learn from this? And I would even say, if another virus were to come up, is there any comments that you guys have on, is this something we can learn? Can we collaborate better in the future? Can we move more quickly? Are there things that you think we are learning from this that can be applied to other diseases, or for future viruses? Is there something you think we're going to have in place in the future that we could react even more quickly to our next pandemic that could obviously come in the future? So for both of you, just to comment on that, and then we could wrap this up.
Yeah,
Rick?
I can go first. I certainly think so. I think some of the vaccine approaches specifically would be the first time that they've been approved to use in humans. So I think that is certainly an area that we're learning about. Being more effective, having more solidified supply chains for our drugs is an important thing that we're learning about. But what we're doing isn't easy. And it certainly is taking resource away from other critical things that we need to be focused on in other disease areas. So I don't think it's as easy as now going forward, we can make a vaccine in a year for whenever we need to. It really is taking a tremendous amount of resource. But certainly there are things being learned that can help us in the future.
Yeah, I very much agree. I think, and Rick touched on this earlier, but supply chain is a critical. As we've seen with diagnostic tests, shortages of swabs turned out to be a critical factor. So learning from that medical glass, we do not have an infinite supply of medical glass for having vials with vaccine, and anticipating going forward, even being prepared for all those components that go into vaccination, not just the vaccine itself, but what it takes to produce the vaccine, get it in vials or syringes, get it transported out. How do you, if it needs to be refrigerated or frozen, being prepared with the supply chain and the distribution chain. So there's a lot to be learned from that. I think looking back at some of the things that have been done successfully in the past, people are now anticipating today and going forward collaborative investigational trials where more than one vaccine is used in a phase 3 study. That was done with acellular pertussis vaccines many years ago and done very successfully with several manufacturers providing the vaccines and then very large scale clinical trials being performed that really helped the timeline very greatly. And that was done with a lot of support and under the direction of NIH years ago. I think those types of things people are considering now. It may not always be what one would want to do, but going forward and in the future, anticipating how collaborative work can go forward. Dr. Nettles has described the collaborations that are going on with J&J for their vaccine and drug products. And in the case of FluGen, we have, I think, a very fruitful collaboration with University of Wisconsin and Barat. So those types of associations will all be to the benefit for getting a safe and effective vaccine out for use.
Great. Well, thank you both so very much. Thank you for working through some of these technical difficulties with me. I really appreciate it. Thank you to your companies for what you're doing, and the other companies out there that are also working on the therapeutics, the vaccines. We truly appreciate it. So I know this has been educational for me, and I'm sure for our audience. So thank you again. We appreciate it.
Thank you, Lisa, and thanks to BioForward.
Thank you.